Hello everyone and welcome to the new session of the lecture series, and today we'll be revising the chapter photosynthesis in higher plants. So we have already discussed this chapter in detail in the previous lectures, and today we'll be discussing some of the topics here. Alright, and rest of the topics we will revise in the next lecture. Okay, so let's start with some of the early experiments. So first is the very renowned name of Joseph Priestley. Okay, so all these experiments are related to the photosynthesis or help us to understand the process of photosynthesis. Okay, so let's continue with the experiment done by Joseph Priestley. So Joseph Priestley, he conducted a bell jar experiment. Okay, why bell jar? Because the jar was bell shaped. That's why it was given the name bell jar experiment. So what he did in first setup, so this is the first setup, he kept a mouse and a burning candle in a bell jar, and then he noticed after some time that the mouse died and the candle exhausted. Okay, it was not burning. So in the second setup, what he did, he kept a plant there along with the mouse and the burning candle, and what he observed, that the mouse was still alive. Okay, you can see in this figure, mouse is still alive and the candle is still burning. So from this experiment, what he showed that the plants are able to restore the air that is consumed by the breathing animals and the burning candles. Alright, so you know that what does plant release? They release oxygen, alright, in the process of photosynthesis, and this oxygen can be used by the mouse and the burning candle. Okay, and then mouse and the burning candle, what they produce? They produce carbon dioxide. Okay, during respiration, mouse will produce carbon dioxide, as well as the burning candle will also produce carbon dioxide. So this carbon dioxide can be used by the plant, okay, in the process of photosynthesis. So from this experiment, what did Joseph Priestley did? He discovered oxygen. Alright, and he said that the plants restore the air consumed by the burning candle and the breathing animals. Okay, so next is the name of Ian Enghouse. Alright, so what was his contribution? So he showed that the importance of the sunlight and green parts of the plant. Okay, you have to remember this importance of sunlight and the green parts of the plant. Okay, he said that green parts of the plants are only involved in the oxygen evolution, and that too in the presence of the sunlight. Alright, so he was working on the aquatic plant Elodea. So this is the aquatic plant Elodea. So in the first setup, what he did, he kept this entire setup. So Elodea plant was there, kept in the water because it is an aquatic plant. So he kept the first setup in low light, or you can say in dark conditions. Okay, and then what he kept the same setup in the bright sunlight or in the light. Alright, and then what he noticed the evolution of the oxygen, okay, in the form of oxygen bubbles. Alright, and from this experiment, he concluded that the green part of the plant, okay, and the sunlight is important for the oxygen evolution. Next is the name of Julius Monsach. Okay, so what he did, he said that glucose is produced by the plants during photosynthesis, which we know is true now, okay, and stored as starch. So storage material in plants is starch. Okay, so now another experiment was conducted by T.W. Engelmann. Okay, and what was his contribution? He gave the first action spectrum of photosynthesis. Okay, you have to remember this action spectrum. Alright, and he also showed that maximum absorption occurs in the blue and the red region of the visible spectrum. Okay, of what? Of the visible spectrum. Alright, so what he did, he was working on the algae cladophora. So cladophora is a green algae. He was working on cladophora. So what he did? He kept this cladophora. So this is cladophora in the suspension of aerobic bacteria. So here all around aerobic bacteria was present around this cladophora. Alright. Suppose this is cladophora, which is green algae. Alright. This cladophora was kept in the suspension of aerobic bacteria. Aerobic bacteria means which utilize oxygen. Alright. And what he did? He kept a prism there, okay? Using a prism. So this is a prism. What is the role of the prism? It scatters the light into its component. So here light was given, and using prism, light was split, okay, into its components. Suppose, and what was observed, that there was accumulation of bacteria here in this region and in this region. Okay. Here there was overgrowth of bacteria. And what were these regions? These region was, okay, blue and the red. So there was the overgrowth of aerobic bacteria in the blue region and the red region of the visible spectrum. Alright, so this is what I have written, that showed that maximum absorption occurs in the blue and the red region. By what? By chlorophyll present in the green algae cladophora. Alright, and from this experiment he gave the first action spectra of photosynthesis. So here in the blue and the red region there was maximum absorption and there was maximum oxygen evolution also. Okay, oxygen evolution also there. That's why there was proliferation of the aerobic bacteria here in these region, because aerobic bacteria utilize oxygen. There was more oxygen in the blue and the red region. So there was more growth of the aerobic bacteria. Okay, so next is the name of the scientist, Cornelius van Niel. Okay, so he was working on the purple and the green sulfur bacteria. So these bacteria, what they use? They utilize hydrogen sulfide instead of water. Okay, for converting carbon dioxide into carbohydrates. Okay, so instead of water, they use hydrogen sulfide. Okay. So now, using this purple and green sulfur bacteria, which are photosynthetic bacteria, he proved that the oxygen is evolved from splitting of water. Okay, means from the photolysis of water and not from the carbon dioxide. Okay, not from carbon dioxide, but from the splitting of the water molecule. Okay, so initially what he did, they were using green and purple sulfur bacteria which use hydrogen sulfide. If you replace A with S, okay, here sulfur will be released, okay, and then glucose is formed and water is also formed. And if you replace this A with O, it becomes water here, and you can see oxygen evolution here now. Okay, you can see the oxygen evolution. And what else did he said? He said that light is very important for the oxygen evolution. Light is also important. Okay. And what else did he did? He introduced the chemical equation of the photosynthesis, which is this: 6 carbon dioxide plus 6 water in the presence of sunlight, okay, forms a glucose molecule and oxygen is released in the process. Now let's talk about the another term, electromagnetic spectrum. Alright. So how can it be defined? It is the range of electromagnetic radiations in ascending or descending order of the wavelength and frequency. Okay, you have to remember this: wavelength and frequency. Alright. So here you can see this is the electromagnetic spectrum here. Alright. First we have gamma rays, we have X-rays, we have UV, and then we have these. This region is the visible spectrum. Okay, so this region can be perceived by the human eye. Visible spectrum can be perceived by the human eye. That's why it is known as visible spectrum. By the human eyes. Alright. Then we have the infrared and then we have the radio waves. Okay. Now let's talk about the visible spectrum. So how it can be defined? The portion of electromagnetic spectrum that is visible to the human eye is called the visible spectrum. Now this visible spectrum has a wavelength range of 400 to 700 nanometer. Alright, this you have to remember. And frequency range from 4 to 10 raised to the 14 hertz to 7 into 10 raised to the 14 hertz. Alright, now let's talk about the absorption spectrum and the action spectrum. Alright, how can we define absorption spectrum? So it is the graphical representation of the amount of light of different wavelengths absorbed by a pigment. Okay, suppose you can take the example of pigment chlorophyll A. Okay, so amount of light of different wavelengths absorbed by chlorophyll A, or you can say chlorophyll B or any other carotene, xanthophyll. Okay, and what is action spectrum? Action spectrum is a graphical representation that shows the effect of different wavelength of the light on what? On a particular biological process, such as oxygen evolution at different wavelengths of light. Alright, or you can simply say that what is the absorption spectrum? It represents the amount of light absorbed by the pigment. Alright, amount of light absorbed of different wavelength, okay, by the pigment. And then what does action spectrum tell you? It tells you, or it represents the overall rate of photosynthesis, which can be measured. How? It can be measured by oxygen evolution. Okay. Now, so this is the graph here. Alright. So this one, graph A. So this shows the absorption spectra of chlorophyll A, B and the carotenoids. Okay, we have chlorophyll A, chlorophyll B and carotenoids. Chlorophyll A, you can see, has the absorption spectrum around 400. Maximum absorption at around 400 and then at around 700 nanometer. Alright. And then let's talk about chlorophyll B. Okay, slightly. Okay, it is similar to chlorophyll A. Okay, here is the absorption maximum. Alright, and then carotenoids. Carotenoid absorption spectrum is different from chlorophyll A and B. Okay, and graph B. What does graph B show you? Graph B shows the action spectrum of photosynthesis. So here you can see there is a rate of photosynthesis measured by oxygen evolution. Okay, which is the biological process. And here we have the wavelength of the light in nanometers. So again you can see here maximum oxygen evolution is here. Alright, maximum peak you observed here. This is the maximum oxygen evolution, which takes place where? In these region around 700. Okay, around 700 and 400 nanometer. Okay. Now graph C. Let's talk about graph C. So this graph is showing the action spectrum, okay, of photosynthesis superimposed on the absorption spectrum. Okay. So this is the action spectrum. This is action spectrum. This is the absorption spectrum. Okay. Here you can see again they are superimposed at around 400 nanometer. You can say maximum absorption of the light and maximum oxygen evolution. And again here this also overlap. Okay, around 700 nanometer. Maximum of oxygen evolution and maximum absorption here. Wherever there is maximum absorption, there will be maximum oxygen evolution. Alright. Now let's talk about the structure of the chlorophyll. So, what the structure of the chlorophyll consist of? It consists of a porphyrin head and a phytol tail. So this is the porphyrin head. This phytol tail is embedded in the thylakoid membrane because it is hydrophobic. Alright, this helps in the attachment of the chlorophyll to the thylakoid membrane. Here you can see there are 4 pyrrole rings. Okay. So again, chlorophyll consists of a porphyrin head, which is made up of what? Tetrapyrrole, 4 pyrrole rings. Alright, so this is the first pyrrole, second pyrrole, third pyrrole, and then we have the 4th pyrrole. So porphyrin is a tetrapyrrole with magnesium atom in the center. Alright, or you can, okay, and then we have the phytol tail. Okay, and this constitutes the structure of the chlorophyll. Now, let's talk about the pigments involved in photosynthesis. Okay, so chief pigment or the primary pigment is always chlorophyll A, which is blue-green in color. Okay, it absorbs visible spectrum of light, means VIBGYOR, and show maximum absorption in the blue and the red region. Okay, and the rest are called as the accessory pigments, which constitute of chlorophyll B, which is yellow-green in color. Then we have carotenes, which are yellow, orange, and red. And then we have xanthophylls, which are yellow in color. Okay. Now, next topic is the type of complexes present in the thylakoid membrane. Okay. So now, the thylakoid membrane consists of 4 complexes. First is photosystem 1. Okay, which consists of the reaction center, consists of a single chlorophyll A molecule with maximum absorption at 700 nanometer. Okay, that's why we also denote photosystem 1 as P700 because the maximum absorption is at 700 nanometer. Similarly, photosystem 2, reaction center consists of again chlorophyll A, okay, and with maximum absorption at 680 nanometer. That's why we write photosystem 2 as like this. Okay, reaction center consists of the chlorophyll A molecule with maximum absorption at 680 nanometer. Now the third complex present is cytochrome B6F. So this complex is involved in the electron transport chain. Okay, and fourth complex is the ATP synthase, which is involved in ATP synthesis. Alright, so these are all present in the thylakoid membrane. Now next term is the light harvesting complex. So this is a diagram of a light harvesting complex. You can see these are the pigments. Okay, there are hundreds of pigments. Okay, hundreds of pigments. So what is light harvesting complex? It is the combination of hundred of pigment molecules bound to proteins. Alright, bound to proteins. So these pigment molecules, also known as accessory pigment, these absorb the photons, okay, energy present in the sunlight, and it transfer it to what? The reaction center, which is made up of what? Chlorophyll A. Alright, and then chlorophyll A gets excited, electrons are released and they are accepted by the primary acceptor. Okay, so what is light harvesting complex? It is made up of hundreds of pigment molecules bound to proteins. Okay, and then let's talk about photosystem 1 and photosystem 2 now. So photosystem 1 absorbs longer wavelength of light, more than 680 nanometer. Okay, and we denote it as like this, P700. Alright, and photosystem 2 absorbs shorter wavelength of light, less than 680 nanometer, and we denote the photosystem 2 as P680. Okay, now again, let's talk about photosystem 1. So reaction center in photosystem 1 is chlorophyll A 700. Means maximum absorption at 700 nanometer, whereas in photosystem 2, reaction center is chlorophyll A 680. Okay, means maximum absorption at around 680. Now, another difference between photosystem 1 2 and photosystem 2 is that there is no photolysis of water in photosystem 1, whereas there is photolysis of water in photosystem 2. Alright. So, next topic is the structure of the chloroplast. Okay. So, chloroplast is a double membrane structure. It consists of outer and the inner membrane. This is known as the stromatal lamella, or also known as the stromatal thylakoids. So what does it do? This stroma lamella connects the two thylakoids. Okay, it connects two thylakoids. Okay, now each one is known as a thylakoid. Okay, and thylakoid are stacked over each other. Thylakoid is stacked over each other. Okay, and then it forms a grana. Alright, here we have the stroma. Stroma is the fluid of the chloroplast. Here we have the ribosomes. Then we have the starch granules and lipid droplets are also present in the chloroplast. Alright. Now, let's talk about the photosynthesis, or the process of photosynthesis, or you can say mechanism of photosynthesis. So, photosynthesis takes place in two phases. First is the light reaction and second is the dark reaction. So, light reaction takes place in the thylakoids. Okay, and thylakoids, they stack over each other to form a grana. Alright, and dark reaction takes place in the stroma of the chloroplast, where all the enzymes are present. Okay, enzymes, catalyst, okay, coenzymes, everything is present there. Again, now let's discuss about the light reaction, also known as the light phase, and the dark phase first. Okay, so light phase, also known as the photochemical reaction. Okay, dark phase is also known as the biosynthetic phase. Okay, now let's talk about the light phase. Why it is known as photochemical reaction? Why? Because the energy from the sunlight is converted into the chemical energy here, or simply you can say light energy is converted into the chemical energy. And what type of chemical energy? This chemical energy is stored in the form of what? ATP and NADPH. Okay. Now these are the product of light reaction. ATP and NADPH are the products of the light reaction, and these products, both ATP and NADPH, are used to synthesize sugars during the dark phase. That's why it's also known as the biosynthetic phase. Alright. Now what happens in the light reaction? Oxygen evolution takes place, and both photosystem 1 and photosystem 2 are involved. Whereas during the dark phase, carbon dioxide fixation takes place. Carbon dioxide fixation into what? Into sugars, or you can say carbohydrates. Okay. And no photosystem is involved. Alright. So what happens? Here we have the light reaction. Light reaction produces what? Produces ATP and NADPH. Now these are utilized in what? In the dark reaction. Okay, both ATP and NADPH are utilized in dark reaction to produce what? Carbohydrates. Alright. Now, next topic is the photolysis of water, means breakdown of water. Now, photolysis of water yields electrons, protons and oxygen. Alright, here you can see this is the water molecule, And what ions are responsible? Manganese ion, chloride ion, and calcium ion are used, and protons are produced, electrons are produced, as well as oxygen. Oxygen is also produced here. Okay. Now the water splitting complex is present in the photosystem 2. That's why oxygen evolution is also takes place at the photosystem 2. Alright. Now what is the significance of photolysis of water? Why photolysis of water is important? So first is that now it is very necessary to replace the electrons that were moved from the photosystem 2. Alright. So now we know that photosystem 2, okay, when it absorbs the sunlight, it gets excited and electrons are released. Okay, so gap is created here. So this gap is filled by the electrons from photolysis of water. Okay, photolysis of water releases what? Protons, electrons, and oxygen. Okay, so these electrons are transported to photosystem 2. Alright, and it fills the gap of the photosystem 2. Okay, now what happens? The electrons needed to replace those moved from the photosystem 1 are provided by photosystem 2. Okay, so similarly, we have photosystem 1. So here what happens? It also gets excited. It releases electrons. Okay, and the gap of photosystem 1, here the electron gap, okay, or the vacancy here is filled by the electrons from photosystem 2. Okay, here electrons are, okay, it gets excited, photosystem 2, electrons are released, these electrons are transferred to photosystem 1. Alright, so electrons need to replace the electrons needed to replace those removed from the photosystem 1 are provided by photosystem 2. Also, what happens? The electron released by the photolysis of water enters the electron transport chain, and this creates a proton gradient. Alright. So what happens? There is a transfer of electrons now, and the transfer of electron from one carrier to other, okay, helps in the creation of a proton gradient. A proton gradient is created as the electron move from one carrier to another carrier in the electron transport chain. Okay. Now let's talk about the electron transport chain. Okay, also known as ETC or the Z scheme. Why it is called Z scheme? Because there is a zigzag flow of electrons. Okay. So what is ETC? ETC is a sequence of protein complexes and electron carriers situated within the cellular membranes that facilitate the transfer of electrons in a controlled manner. Now the electron transport occurs in a zigzag manner from molecule with low redox to high redox or reduction potential. This you have to remember. Now next topic is photophosphorylation. How it can be defined? Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. This you have to remember. So the energy stored in the light is used for the synthesis of ATP. Okay, now photophosphorylation is of two types. First is cyclic photophosphorylation. Second is non-cyclic photophosphorylation. Let's discuss the cyclic photophosphorylation word first. Okay, so what happens in cyclic photophosphorylation? Only photosystem 1 is involved. Now the electron ejected from the reaction center of photosystem 1 return to its reaction center. Okay, back. That's why it is known as cyclic. Okay, and there is no oxygen evolution here because oxygen evolution takes place where? What is the site? Photosystem 2. Okay, but here no photosystem 2 is involved, only photosystem 1 is involved. Okay, now the first electron acceptor in cyclic photophosphorylation is a FRS, ferredoxin reducing substance, which is an iron sulfur containing protein. Okay, earlier ferredoxin was considered as the first electron receptor, but it is not. Okay, ferredoxin is not the primary receptor. Primary receptor is FRS, ferredoxin reducing substance. Okay, and then electrons are transferred from FRS to ferredoxin. Okay, now this is the flowchart of the cyclic photophosphorylation. So what happens? Photosystem 1 is only involved, which has the reaction center of P700. Okay, single chlorophyll A molecule is present as the reaction center. It gets excited, okay, by absorption of the photons. Electron receptor is present here, all right, which is an FRS. And what happens? Again, the electrons, they go back, okay, or they cycle back to the reaction center again, and in the process ATP is produced. All right. Now let's talk about the non-cyclic photophosphorylation, which is also known as the Z scheme. Okay, here both photosystem 1 and photosystem 2 are involved. It occurs at the grana thylakoids only. Okay, this you have to remember. And then what happens? The electron ejected from photosystem 2, okay, never goes back to the photosystem, okay, to the chlorophyll A, never goes back to the reaction center back. That's why it is known as non-cyclic. All right, and it's finally accepted by the NADP plus, and NADP is converted into NADPH. So during non-cyclic photophosphorylation, both ATP and NADPH are produced. Both. All right, both ATP and NADPH are produced. Okay, oxygen is involved. Yes, because photosystem 2 is involved here. And here pheophytin is considered as the primary acceptor of the electrons. So this is the flowchart of non-cyclic photophosphorylation. Here you can see this is photosystem 2, which contains light harvesting complex 2, you can say. Here for LHC1. And this is photosystem 1. Okay. Light, both of these photosystem absorbs light, they get excited. Here electron receptor, primary electron receptor is a pheophytin. All right. Then what happens here? Electrons move downhill. Okay, electrons are moving downhill. Okay. Here again it gets excited, photosystem 1. Okay, here the gap, electron gap is filled by the electrons from the photosystem 2. Alright, and here the gap is filled by the electrons produced by the photolysis of water. And here you can see both ATP and NADPH are produced. Okay, and the electrons never go back to the reaction center but is accepted by the NADP, which gets converted into NADPH. Okay, now let's talk about the chemiosmotic hypothesis. Alright, so chemiosmotic hypothesis was given by Peter Michel in 1961. And what he proposed? He proposed that the ATP synthesis. So he was talking about the ATP synthesis, how ATP is synthesized. Okay, so he proposed that ATP synthesis is linked to the development of the proton gradient across the thylakoid membrane. Alright, so here this is the diagram showing the chemiosmotic hypothesis. So what happens? Photosystem 2 is present in the thylakoid membrane. This is the thylakoid membrane. Photosystem 1 is also present here. Then we have this complex cytochrome B6F. Okay, and the fourth complex present is ATP synthase. Alright, so what happens as the electrons flow through the electron transport chain? So what happens? The proton gradient is formed here. So this is the stroma. Okay, so the protons from the stroma, they move into the lumen of the chloroplast. Okay, here you can see hydrogen ions are accumulating here. As there is the flow of electrons, the hydrogen ion accumulate in the lumen of the thylakoids. Okay, and what happens then? What happens here then? There is a breakdown of the proton gradient. First, there is a development of the proton gradient. Okay, development of proton gradient. All right. And then what happens? Second is the breakdown. Breakdown of proton gradient. All right. And then what happens? Due to the breakdown, how does breakdown of proton gradients take place? So this is the complex is known as ATP synthase, and it has two parts. All right. So this complex is known as ATP synthase. It has two components. First is CF0, and this is known as the CF1. So CF0 acts as a transmembrane channel. Okay, so CF0 acts as a transmembrane ion channel, okay, through which the protons flow back to the stroma. Okay, and what happens as the protons flow back to the stroma here? Okay, they pass through F0, and there is a conformation change in the F1 part. All right, there is a conformation change, there is a rotation, mechanical rotation, and this leads to the formation of ATP, or you can say this creates the energy for the formation of ATP. All right, so this was the chemiosmotic hypothesis, which explains the synthesis of ATP. All right, so this was the end of this lecture. These are some of the references. I have mentioned some of the references. First is the NCERT textbook, and these are some of the sites. Okay, you can refer to them. So, thank you very much for listening. We'll meet again in the next lecture. Thank you very much.